Now let's look at the structure of organelle that is ultimately responsible for converting or translating the nucleotide language into the language of amino acids. So as we know, the organelle is ribosome. The ribosomes have two subunits. These subunits exist independently when they are not engaged in translation or formation of the protein using messenger RNA. The large subunit in eukaryotes has 3 RNA molecules and 45 polypeptides or protein molecules. That's the composition of the large subunit. The small subunit has 1 RNA molecule and 33 polypeptides or protein molecules. The interesting thing about this organelle is that it is the whole complex self-assembles. So if we take these individual components, 3 ribosomal RNAs, 45 polypeptides, and put them in a solution, they will form the large subunit of the ribosome. This is free energy at its work. The delta G, remember we talked about earlier in this course. So all systems try to come to configuration which is the least energetic form possible. The forces that are keeping these particles together are ionic and hydrophobic. In this complex, the large subunit is the one that is responsible for catalyzing the reaction and forming the peptide bond. We'll look at that later. So here I would like to mention a few more facts about ribosomes. They can use any messenger RNA. Ribosomes are not specific for specific messenger RNA. A ribosome, given ribosome, can bind, interact with any messenger RNA and result in production of the relevant protein. So these ribosomes of prokaryotes and eukaryotes have some differences. So this fact has been exploited by scientists so they can come up with agents that are disruptive to the ribosomes of prokaryotes or bacterial cells and not to the eukaryotic or our cells. So there are many drugs, for example tetracycline. It affects the ribosomes of the prokaryotes or the bacterial cells but not the host cells, which is us. Mitochondria and chloroplast, I have mentioned this fact before too, also contain ribosomes, and these ribosomes are similar to the bacterial ribosomes rather than the eukaryotic ribosomes, the cells in which they live. The large subunit has 4 sites. Here I would like to point out that some books, some texts only mention 3 sites, but since our textbook has 4, I'm going to proceed with the 4 sites. T site, the transfer site, first site where the charged tRNA lands with the help of a transfer factor. The amino acid site or the A site, here the anticodon binds to messenger RNA codon. P or polypeptide site, here this is the site where the reaction takes place and the formation of peptide bond is formed between 2 adjacent amino acids. The E site or the exit site, after a particular tRNA has donated its amino acid, it leaves at this E site. So we have looked at the structure of these organelles. Now next we will see how they function.